Presented by Caltech.
It is a great honor for me and Chifu to join you to celebrate the new adventures and new directions that are taking place right here. It is really awesome to be in this glowing brilliance that's reverberating in this room. My God, megawatts. And I came to Caltech in the fall of 1978. I remember clearly during the first week of classes, seminars, and working in Jorgensen's basement, furnished with a bunch of beanbags. I thought to myself, wow, these are really happy people. They crack jokes, laugh their hats off, amid these animated questioning and discussions. The only problem was I couldn't get the jokes. The best I could manage is to pretend and follow their laughs. Fortunately, I had a few helpful fellow grads. Phil, the Cookie Monster, among them. Anyway, they gave me generally helpful tips, such as what TV shows and sports games to watch and the music to listen to. But critically illuminating, they recited a list of Carver's puns. And so Carver, our beloved punster, created so much fun for us amid in an otherwise high-octane environment. In fact, in a couple of hours, all of you are going to be deserted at a coffee break. Get it? Get it? Okay, all right, back to business. Let me now just turn to just one aspect of the global economic impact Carver's work has brought forth. Is it a coincidence that the Taiwanese semiconductor foundries have a combined world market share of around 60%? Okay, all right, okay. So the time was early 1980 when the Taiwan's Hsinchu Science Park, aimed at emulating Silicon Valley, was newly established. At the invitation of Mr. K.T. Lee, who decades later was adoringly called the father of Taiwan's economic miracle. Carver met face-to-face with Lee, as well as visited the Industrial Technology Research Institute, ITRI. There, Carver's gospel of free entrepreneurism and the Silicon Foundry was heard loud and clear, and wholeheartedly believed, nurtured, and executed. And the rest is history. Okay. All right. Over the years, I appreciate ever more the dynamic ranges of Carver's work, still going strong, way out there, and way down into the atomic scale, such as superconductors. As well, Carver has this take no prisoners approach to managing complexity. So only through clarity one can build and truly understand. So thank you, Carver, for such powerful intellectual demonstrations that really enriched my life, and they have also on many occasions nudged me to shift my vantage point, to alter my perspective, take risks, and expand my horizon. So today's celebration highlights the innovations that are inspired by Carver's spirit of fearlessly breaking new ground. The first speaker of the session is Professor Shuki Bruck. Shuki is the, he sits in Carver's old chair, the Gordon and Betty Moore Professor of Computation and Neural Systems and Electrical Engineering, on and on. Okay, all right. So, okay. Now I need my cheat sheet. Okay. Shuki joined Caltech in 1994, and in 2002 initiated and led the formation of the Information Science Technology Program, IST, and served as the founding director. Shuki created the first Caltech IST undergraduate class, IST-4, in 2007, and taught it since. His teaching was recognized by the 2009 Richard Feynman Prize for Excellence in Teaching. Shuki's entrepreneurial experiences include founding and serving as chairman of Rainfinity, Extreme.io, and MemVerge. His talk today is about a new approach to fighting cancer. Cancer emerges from the natural accumulation of errors, somatic mutations, that occur in the normal processes of cell division. He hypothesizes that the estimation of the number of somatic mutations in healthy cells can serve as an early indication for an individual's susceptibility to cancer. Affirmative answer to this hypothesis will enable affordable screening as well as early detection protocols that might dramatically change our approach to combating cancer. So, Professor? Oh, Carver.
Thank you, Marina. Thank you, Shifu. Yeah. Yeah, I love Caltech, and I feel very fortunate to be here. Definitely in events like this, it feels like a Passover dinner. I'm Jewish, or Thanksgiving. So, thank you for including me. If I was to write a proposal on cancer, I never worked on cancer. I never worked with DNA data. I'm not sure if I can write more than a few words in applying for funding. Probably I would write, I'm ignorant about cancer. However, I'm enthusiastic outsider. Yeah. So what I want to do today is not just tell you what we accomplished, but also, because it is a completely new area for me, also to tell you what is a thought process. How did I get to where I got to? Because it's still obvious and still very fresh in my mind. So the reason for what I'm doing is that the likelihood for a person to develop cancer in their lifetime is about 40%. And that's an underestimate, because if you look at data from the UK, it's close to 50%. If you assume that we will live longer and take care of many other things, it will start getting close to 70%. So my conclusion is that cancer is just part of life. Life is also a dangerous disease. So it's more or less in the same category. So what is life? What is life? It's an information system. In fact, it's the first information system that ever emerged in the world, about 3.7 billion years ago. So in order to explain to you my view on life, I will tell you my life story. Now, this is not my life story, is my life story. So life is a bunch of funny shapes that have a comic sun in the middle. That's how I think about life. And those funny shapes, they move around, they are diverse, and they are complex. And sometimes they replicate, so we have more funny shapes with comic sun in the middle. Then some of them get tired, and they get extinct, so we have less funny shape with comic sun in the middle. Then we have some kind of innovation. We suddenly get new shapes that are moving around, and some of the shapes disappear, and there is a process of selection, and we get completely new shapes, maybe a little bit boring shapes. And that is life. This is my life story. In fact, about 200 years ago, a person by the name of Darwin went to a very exciting place, the Galapagos Islands, and he figured out that this whole process that I discovered to you at the macro level can be explained by the concept of evolution. It took him about 25 years to write a book about that. That's the origins of species. And that's where our state was up to about 100 years ago, or maybe less. The question is, what is this I, the memory in the middle of the funny shape? And the person that addressed that is someone that I think would qualify for the Carver Mead Fund, is Schrödinger, that was a physicist who won a Nobel Prize in the 30s. And he was sitting in his office thinking, what is really life? What is this memory of life that is in the middle of this funny shape? So being a physicist, he used physics. The way he looked at life, he thought, well, life is small, life is precise, and life is diverse. So those are the properties I can see from the outside. Now, being a physicist, I can think about two ways to implement this I, this memory. The first one is using particles that interact and create some collective phenomena. That's my first idea as Schrödinger. However, the problem is that given the size and the required precision, it's not possible to do it. He did all the computation to actually prove it. So what is the other option for a physicist is a crystal, a periodic crystal. This one is precise. The problem with this one is that it's not sufficiently diverse because it's boring. It's repeating. Okay, so he took a walk and he said, I actually don't have any good idea using the concept of physics to explain life. That's exactly the point that the concept of information was born. That's exactly the point. Notice this is 1944, before we had computers, before Shannon wrote his information theory paper, and many other things that didn't happen. His idea was, let's encode the information using an aperiodic crystal. In nature, we don't see aperiodic crystal unless it's life. In fact, this is text from his books that I highly recommend. He talks about a miniature code The number of atoms in such a structure need not to be very large to produce an almost unlimited number of possible arrangements. He didn't know anything about the genetic code, the fact there are 4 symbols, that there is double helix. He didn't know any of that. He just did an analysis from the outside using the concept that he understood and discovered that we must have something like that, and he was right. Unbelievable. Fast forward. Fast forward, we can read. We can read the book of I. We can read the memory of life. And now, if you spend about $100, you can get to you can read your own book. Kind of boring. I did that. Both long and boring in my case. 3.2 billion base pairs. The other thing we learn is that we replicate the memory, there are errors. And when we say errors, it sounds negative. But in fact, errors is the reason we exist here. Otherwise, we will not be here. Errors is the foundation of innovation. That's how we create new things, and then through a process of selection, we get better things. What kind of errors? I'll show you a couple of examples. The first type of error is just a typo. We copy, and instead of a letter C, we get a letter G. There are many other errors. Another one, interestingly enough, is an error of duplication. We copy, and then whoever makes a copy falls asleep and copies something twice next to each other. That's called tandem duplication. Interestingly enough, 50% of the DNA is duplications. So Schrödinger was not exactly right, because he thought that it will be aperiodic. About half of this is kind of periodic. Cancer is caused by mutation. The same mechanism that creates innovation is a reason for cancer. Same mechanism. What are the reasons for cancer? What kind of reason lead to mutations? The first one is heredity. In this case, when you have errors, you should blame your parents. You know, some people like to blame their parents, but this is another case. The second one is environmental. Smoking, doing something not right. In this case, you blame yourself, not easy, or you blame the government. That's easier. The third one is random mutations. That's how it works. That's a mechanism of evolution. Here you cannot blame anyone, but I think the key is that that's a recent paper from 2017. Turns out that the most dominant reason for having cancer is random mutation. The weakest one is heredity. So I recommend to everyone, don't blame your parents. And of course, environmental. So given that, again, I'm walking in my ignorance lane and holding Carver's hand while I'm walking in my ignorance lane. So I got to this point and I say, okay, you know, about half of us will get cancer in our lifetime. What is the current approach for dealing with cancer? It's completely reactive. We deal with it only when it happened. Assume that you have a car that you know will fail with probability 1/2 within the next 2 years, are you going to wait for it to fail? Probably not. We do it, all of us, with cancer. However, it's not it's not like catching a virus, cancer. It is a continuous disease. It happens over your lifetime. The question is, can we do anything about it if it's accumulating? Or should we wait to a point where it emerges? That's a question I ask myself. I ask, can we measure and quantify mutation accumulation? This is in healthy people. That might lead to some more active approach. Completely different approach for treating it. Right now we don't do it. How will we measure mutation accumulation over time? So the ideal way is to, when someone is born, is to take a blood test, analyze the DNA from the white blood cells, do it later on, do it. The person gets to 97. In this case, that's the age they got cancer, and we can learn from that. By the way, it takes 97 years. This data right now is not available. It's something we should do. We should do it not just for cancer, but we should do it for many other things. Because many diseases are a result of mutation accumulation. So the question is, what should we do now? That's a question I asked myself about a year or two ago. We cannot do the blood test of over 97 years. So I want you to help me find a solution. Here are two pictures. The top one kind of looks random. The lower one doesn't look random, looks like a chessboard. What I ask you to do right now is close your eyes, and I will introduce an error into both of them. And when you open your eyes, I want you to tell me where are the errors. So please close your eyes. Now you can open your eyes, and does anyone see an error in any one of those? You see it in the lower part or upper part? Lower part. So in fact, the same error happened in the upper part. And here is another error. What is the conclusion from what you just advised me? Is that it's very easy for us to detect errors in areas that have some pattern. That's a very simple idea that we came up with. Here is an example from chromosome 1 of humans. It's a big repeated area. It's not exact. You can see the errors. How do we know the errors are there? Because they are in an area that is repeated. We are also very lucky. Why are we lucky? There are about 700,000 areas of repeated DNA in a human DNA. There are about 10,000 of those in the area that is a coding area, the exome. So we have many places where we can do those measurements. The other lucky part is the likelihood of developing a mutation in a short tandem repeat area, that's how they are called, is 10,000 times what the probability of developing a mutation in an area which is not a short tandem repeat. So in some sense, nature gave us a set of antennas, speaking in the language of electrical engineering. 10,000 antennas, or maybe 700,000 antennas, where we can detect activity of mutations. That's a very important aspect. Why is that? Because now we can estimate the mutation accumulation in an intrinsic way. We don't need to look over generations. We can see how the person's own DNA is changing over time. That's a gift that we received. The question with that is, say I do it and I measure it and I discovered this is a signature over 10,000 regions of mutations for a given person. How do I know how it will relate to cancer? I have no idea, because I don't have the time to wait until the person is 97. So here is a second idea. The second idea is, how about if we quantify that, not in healthy people, but we quantify it in non-tumor DNA of cancer patients? Because that's the only thing we have. It's not that it's ideal, but that's the only thing we have, is we can take, say, cancer patients that they have glioblastoma, brain cancer, look at the signature of their mutations in the white blood cells, and see if it's somehow associated with the fact that they have GBM. So all of what I'm describing to you is myself and my students sitting in an office and brainstorming. At the end of the day, all of those ideas are very nice, but we need to see if anything will give when it comes to working with the data. So if I wrote a proposal to the NIH, that's what I want to do, I'm not sure they will even open the envelope. From the outside, they will see my name and will say, okay, send it back to the sender. We worked with data from TCGA, it's the main database of cancer patients in the NIH. It has about 11,000 patients, 33 cancer types. And interestingly enough, they have both healthy DNA from blood cells, white blood cells, and tumor DNA, as well as other data. The reason they have the healthy DNA is so they can compare it and figure out where are the mutations in the cancer. But we are not interested in the mutation of the cancer. We are interested in the process that led to it, which we might get an idea about in the white blood cells. So I'll show you an example with two cancers for which I think everyone knows about it. It's clear that it's mainly related to luck. First one is brain cancer, GBM, and the second one is pancreatic cancer. For both of them, we analyze mutation accumulation parameters. And now data science comes in. I was asked to give a Scientific American talk, so I have no equations and nothing that looks scary. Actually, it's fun. What we did is, given those examples of cancer patients that have either brain or pancreatic cancer, we used an approach to build a classifier to distinguish between them based on the healthy DNA. And we build a classifier. A classifier is a software or an algorithm that, given the mutation accumulation profile, or parameters, can tell you what is more likely, that it's a GBM patient or a pancreas cancer patient. So we did all of that. And of course, one option is that we will do it, and we will get that when we introduce new patients that we did with pancreatic cancer, we'll get 50% likelihood. Brain, 50%. in this case, I'll send Zita and Adam an email saying, we got stuck. I don't know what to do right now. We'll figure it out. However, for pancreas, we got at 84% likelihood and 16 for brain. And when we put brain in, we got even a more striking result that is very clear, that we have a signal. The signal means that we can keep going. It doesn't mean more than that. We are not doing those tests. We are not doing anything. It's just a way for us to say, okay, we can keep going and keep studying it. Of course, we did much more than that. How am I doing with time? I'm fine. We looked at about 5,000 patients, 11 different cancers. We broke them down to brain, skin, and pancreas, and 8 other, and we build like a profile of likelihood. That's the likelihood when we introduce GBM brain patients to the 4 classifiers. A very strong signal for brain. For skin is not as strong, but still is pretty good. Pancreas is still okay, but not as great. And those other 8, it's, I wouldn't say there is a signal here. So it's all preliminary. It's all very preliminary. I think the key message is that there is a cancer-related signal in the blood DNA of cancer patients. That's the only statement I can make. And of course, there are many other questions. You know, we dealt with not with healthy people. We should do that. Why is it working for some cancers and not other? Is the signal related to heredity, to development, to something that happened during lifetime? We're still working on that. Looking at different tissues, not just white blood cells, whole genome versus exome. What will be the clinical approach if it all works? Also not clear. I talked to a number of oncologists about that. Other diseases. There are many diseases that are a result of mutations. Can we use it in other diseases? And in general, the aging process. It's a process of accumulation of mutations. People, you know, someone asked me, how do you know there is an accumulation of mutation? I said, take a look at the mirror. I want to thank you all, and especially thank my team. Siddhar Jain just got his PhD, oh, not next Friday. We'll get a PhD from the president. Bijan is a young student. Siddharth is a EE student. Bijan is a CMS student. And Nathaniel is a postdoc in CMI, starting a faculty position in a few months. We have a paper on that in the bioarchive that has many, many details. And I welcome you to take a look at that. And I'll be happy to take any questions.
Questions? Please come to the microphone.
Thank you, sir, for a very interesting topic. What I don't understand is, if you took my DNA and based on that you said your propensity to get cancer based on data science, what can I do with that? I mean, it's you're not telling me anything about what I should do to change the course. I know you pointed out the end of it, some of the ongoing thing that need to be done, but can you shed some light on your thoughts about what can somebody, how can somebody use this information?
Thank you. That's an excellent question. The short answer is, I don't know. The longer answer is, right now, the reason you ask this question, and I do, and many as a people is because we have an industry that is set up to be reactive. So we invested billions and billions of dollars to be reactive. You go to City of Hope, if you have cancer X, they say, okay, here is what are the options. If you go to City of Hope and tell them, it seems based on what this guy at Caltech is saying, I might get glioblastoma in 5 years. And they say, okay, we don't know what to do. So I went and talked to oncologist, actually an expert in glioblastoma, and he said, actually, I think if we spend time on that, maybe we can develop an approach using drugs to block the transition to a certain state in glioblastoma. Now, I'm not an expert in that, but what he explained to me is that glioblastoma goes through different stages, and there is a stage that is very hard to treat it once it gets there, and it happens very fast. So if someone can both get screening and get some prophylactic medication to make sure that it doesn't jump to the crazy stage, then maybe it's a solution. And in fact, they do have a drug that can deal with it in a very early stage. So that's just a simple example, but we didn't invest in this area at all. We did not invest. And something that will happen to all of us, maybe we should find a way not to wait until it's happening.
Professor Bruck, great talk. Do you have any way of guessing or estimating what the false positive rate might be if you tested a normal population, even for brain cancer?
Yeah, so we have both specificity and all of those results, but as you can see for brain cancer, the false positive is, I would say, it depends what you're comparing to. It can be up to 15%. But again, it's just a beginning. Our goal was not to come up with a perfect way to diagnose cancer, but to see if there is a signal. In fact, there are papers coming out showing that there is a signal on accumulation of mutations in certain tissues before cancer is developed. Actually, there is a science paper from this week on that. By the way, I went to the, I was brave. I'm very proud of that. I went to the NIH in March, to the National Cancer Institute, and I gave a talk on that, 1-hour talk. And I told my students, maybe you should wait under the window in case they throw me and you catch me. It took like 2 hours. All the experts were there and they liked it. So we might collaborate with them. But those are very simple ideas, what I'm presenting here. It's a different paradigm. We still need to do a lot of work. Yes, please.
First, thank you. So based on your results, did you train your classifier on data of each patient or a pool of patients? I mean, is this kind of cancers patient specific or no? So, the reason that I'm asking this is that, for example, if we see a special pattern in the blood of people with cancer, is it possible to generalize that to other people and say that, okay, if we see this kind of pattern, then for other healthy people also, if we see this pattern, it means cancer?
I just will repeat what we did. We trained it on cancer patients. We tested it on cancer patients that we didn't touch during the training, right? That's all what we did. We didn't do it on healthy patients. We did work trying to understand the mechanism, to try to understand what is special about the repeat regions that give us a signal. It's still ongoing. So we do a lot of that. So we didn't discover this pattern that you're alluding to. And we didn't try it on healthy patients.
No, my point is that actually, are you training on the same patient and testing on the other data of the same patient, or you are training on multiple patients and testing on some other multiple patients?
The second one.
Second one, okay.
Yeah.
Okay.
Is it you approve of this approach?
I want just to...
No, I'm never cynical. People that know me, I just speak slowly, but I'm not cynical. Hey, any of you, whether you feel comfortable to speak right now, or you are willing to speak to me one-on-one, or send me email, you are welcome to do it, because we need help. This is a serious issue. Any idea, come to my office, we'll start talking about it.
Sure. Thank you.
All right, we need to keep the symposium going. And any more questions, please come afterwards. Okay, thank you. Thank you very much.